Hi, you guys. I gotta move this over real quick. I wanted to talk a little bit more about membrane transport. I know we did membrane structure in class, but membrane transport is a part that we only did a lab on, and I didn't get a chance to explain other than passive, the different forms of active transport, or co-transport, and another form of passive transport besides osmosis, which is facilitated diffusion. So we talked about passive transport as basically moving from high to low with a concentration gradient. There is no energy required in order to do this. Molecules will naturally move that way. Small molecules and nonpolar ones will be able to diffuse through the membrane easily. So this is a couple pictures from your book illustrating that concept. We talked about osmosis as the diffusion of water specifically through down its concentration gradient through a biological selectively permeable membrane. And then we get words like hypotonic, isotonic, and hypertonic. I want to make sure to use these words appropriately, and I want to see that you can use these words appropriately as it applies to the lab that we did with the potato cells. But here's an example from your textbook using a few other types of cells and how we would refer to these words. Hypotonic is a descriptor. It's a way to describe a solution in terms of its solute concentration. It is always in response to another solution on either side of a membrane. So it doesn't have to deal with it being inside or outside a cell. It's just on either side of the selectively permeable membrane. We say it is hypotonic relative to the other side whenever it has a lower solute concentration. Hypo means low, right? So whenever the solute is low, that means that there's a high water potential. So water will move from high to low. So if the hypotonic solution, if that means that there's a lot of water in the solution and inside the cell relative to that solution is hypertonic, water will move towards the hypertonic solution. It will move to where all the solute is, where there is lower water potential. And you see that in the picture on the left, in the left column, where you have a blood cell that is actually lysed. It's exploded because water is continuously moving in towards all of that solute, trying to go from high potential, where it is outside, to low water potential inside that cell, eventually causing it to rupture. Plant cells don't do that because they have a rigid outer cell wall, but having a very full vacuole helps keep them turgid, which is responsible for turgor pressure and keeps the cell rigid and allows it to have more structure. Isotonic is an environment in which both sides of the membrane have equal solute concentrations. Therefore, water will move both ways through the membrane at equal rates. And then hypertonic is whenever there's a really high solute concentration. If that is the case outside the cell, water will then move out towards that hypertonic environment, and it can cause a living cell to shrivel up or a plant cell to wilt. Facilitated diffusion falls under the category of passive transport as well. It just makes the use of transport proteins so that substances that have a hard time crossing the hydrophobic barrier or the nonpolar region helps them get through faster. They provide a buffer or a channel or opening so that these molecules can get carried through faster. This is a picture of facilitated diffusion with a carrier protein and a channel protein. There's the channel one is to the left, and that's the one where the triangles are going through. This protein has basically an open tube that just allows the free flow of those, maybe they're charged particles like ions, or maybe this channel protein is an aquaporin. It's a specific protein for water to move through quickly. And then you have a carrier protein there on the right. A carrier protein is one that has to interact with the molecule in order to get it across. So there's, again, two ways. You can provide a hydrophilic channel, like the channel protein, or you can bind and carry molecules across like a carrier one. Again, you notice the blue circle has like a little active site on the protein where it seems to have matched its shape. So carrier proteins are usually pretty specific, and they're meant to fit a certain type of molecule or ion that it is responsible for transporting. This is how we get sugar into our cell. I did mention aquaporins a second ago. That is a channel protein that is open for water. Cells in your kidneys have a high percentage of aquaporins in the regions where you want maximum water absorption. So we keep them open and water can move through really, really quickly. That's what you see in the picture down on the bottom right, is a cross-section of a nephron in the kidney. Transport proteins for sugar. Again, these are carrier proteins. You can see open to the outside environment in the picture on the left, and then glucose binds. It causes a shape change in the carrier protein, which makes it open up on the other side and allows it in. These are glucose transporter proteins. And then we have active transport. Active transport is going to require ATP on the part of the cell. So the phosphate, or the ATP, the triphosphate, will bind with those specific proteins. It will break that phosphate off, and the energy released will then cause a shape change in the protein, allowing it to open or close, either to the inside or the outside. This is useful for transporting molecules against a concentration gradient. Now, why would you want to push things to a side where they don't want to go? A good example of this is our sodium-potassium pump, and this is how we create proton gradients. You can push a whole bunch of molecules to one side of the membrane. They want to go back in, and you're going to let them back in, but only when you say so, which will create or allow the cell to do work. An example is, again, this electrogenic sodium-potassium pump that can generate a voltage across the membrane. We're going to pump a bunch of sodium out, but we're going to pump more sodium out than potassium in, which means you will have a slightly more positive charge on the other side of the membrane, and on the inside is slightly negative in comparison, creating a charge difference. This is how nerve impulses are conducted, is we create this difference and then we open the channels up and allow them to swap places, and that creates an electric current down our nerve cells. Another example, and we're going to see lots of this next week, this is a proton pump. Proton pumps are used to push hydrogen ions across the membrane. You can push them out in high amounts and then allow them to flow back through, but only through a protein that does work for the cell, like ATP synthase in the mitochondria, and so that you can produce ATP with the movement of these protons. It's a small energy input in order to pump them, but the energy you get out of their movement is going to be greater. So on the part of a cell, this is a benefit. Makes a lot of sense. Co-transport is when we will allow them to move back in, but we're going to have them carry a solute particle with them. An example in the book is the co-transport of sucrose using a proton gradient. We're going to pump the protons out into the extracellular space. We are going to allow them to come back in through a different type of protein, one that can connect to both a proton and a sucrose molecule at the same time. And then they're going to enter the cell together. And this is the way that plants can store sugar, is because all these protons are going to want to go back in really, really bad because we created a concentration gradient. And every time they do, they're going to grab a sugar and pull it in with it. So even though there might be a lot of sugar in there, we're going to keep adding more sugar so that the plant can live through hibernation. Like in the fall right now, the leaves are storing excess sugar for the winter, and they're using co-transport to get that done. So in review, passive versus active. passive energy or passive transport requires no or very small amount of energy versus active requires ATP to move things. Passive moves molecules from high concentration to low concentration versus active, we're going against gradients. Down versus against. And then our examples, passive transport is diffusion and osmosis, which is just diffusion of water, facilitated diffusion with the help of a channel protein or carrier protein. And then our examples for active transport were sodium potassium pumps, and I'm going to give you endo and exocytosis in a minute. Here's picture examples of the different types of transport. Make sure that when you look at this diagram, you can explain what is happening in each one and why they are good examples. We talked a little bit about osmosis and the importance of cells in order to balance their water. Single-celled organisms like paramecium or amoebas, they have to have ways to balance their water because they might end up in an environment where the solute is not the same or equal as them. For example, freshwater organisms live in a very hypotonic solution. They live in an environment where there's very low solute, but inside their cells, they have so much stuff that they're making and using. So in order to prevent the water from flowing in and making them burst, they have vacuoles that can contract, that have really cool microtubules on them that help force the water back out so that they can stay the same size. And this is the active transport, endo and exo. This is a bulk transport, moving large amounts of materials into and out of a cell. So we can transport lots of proteins or carbs or any other large molecule in, and we can get rid of them quickly and easily as waste. So this is the using vesicular transport or the formation of vesicles. It actually takes away membrane from the cell. The cell gets a little bit smaller whenever it takes in material and makes membranes from its outer membrane. But remember, it's also doing exocytosis at the same time, adding membrane from the vesicle system, the endomembrane system coming from the Golgi. So this is, again, a dynamic structure. It's constantly taking little pieces away and adding new pieces of membrane all the time. Endo means into, think exo means exit or leaving. This is pictures from your book, just showing the bringing in of a bunch of materials from the outside. This is how a lot of single-celled organisms get their chemistry done. They move massive amounts of materials into and out of. Phagocytosis, phage, that prefix means to eat or eating. So this is a eater cell. Cyte, that means cell. Pino has to do with drinking or liquid. And then we have receptor mediated, which means it only happens whenever there are things locked on to the receptors. Then that causes the membrane to engulf those substrates. And we did water potential in class. This is the exact same PowerPoint that I went through with you to teach water potential. So it is available for you to look at on the website, but that's it for the lecture for this time. So you guys have a great afternoon. I have to end this now. I just don't know how.